Okay, great. Thanks for inviting me to come speak to you about my DPhil research. I'm going to go over the goal of my research, what I've accomplished so far in the first year, and what I hope to accomplish in the coming years. And any feedback or advice you guys have would be greatly appreciated. So the goal of my research is to develop a point-of-care assay to detect antimalarial drugs from fingerstick blood samples. So the current first-line therapy for Plasmodium falciparum malaria are artemisinin-based combination therapies, or ACTs, in which a fast-acting and fast-clearing artemisinin derivative is paired with a slower-acting and slower-clearing partner drug. And part of the rationale behind the combination therapy is an effort to avoid the emergence of resistance against artemisinin derivatives. So if any parasites survive the quick and potent dose of artemisinin, there remains partner drug in the patient's blood to remove these remaining parasites. So this eliminates scenarios in which any parasites are exposed to artemisinin but not killed. But despite efforts to prevent the emergence of resistance against artemisinin, parasites with slower clearance rates in the presence of artemisinin have been isolated in Cambodia and spread through other countries in Southeast Asia. And as resistance to artemisinin increases and spreads, more parasites are exposed to the partner drugs of these combination therapies, and you start to see reduced sensitivity to the partner drugs as well, leading to a complete failure of the ACT, at which point the World Health Organization recommends that patient's parasites be treated with an alternate ACT, so a different combination with partner drug. And in a recent study, one of my PI's clinics in Cambodia, they found that 40% of malaria patients presenting to these clinics already had detectable levels of partner drug in their blood at the time of presentation, so likely from a previous treatment, and that these patients were 4 times more likely to fail subsequent treatment with the same ACT. So this suggests that the baseline partner drugs detected in a patient's blood could indicate that they've already taken an ACT with that partner drug. It failed to kill their parasites, and next they should instead be treated with an alternate ACT. However, methods for determining previously failed treatments, methods for detecting baseline drug at the point of care are limited. So they include patient recall, asking the patient what they've taken, but we find that often patients aren't able to definitively tell you which ACTs they've taken. Health records, but if the drugs were obtained at a pharmacy or a distribution center, there are no health records available. And then the gold standard method for detecting blood drug levels is LCMS, but this requires collecting a sample, shipping it to a central lab, and the results can be obtained a couple of days later at the fastest. So this clearly isn't a point-of-care method. So the goal of my research is to develop a point-of-care assay to detect these most commonly used partner drugs from a fingerstick blood sample. And an assay like this would allow for the identification of previous treatment failures. It could be used to monitor compliance for mass drug administration campaigns, and could also be used to generate real-time maps of drug use. The idea for the assay is a lateral flow paper-based assay where whole blood can be applied to one end of the assay, and plasma can be isolated in a secondary detection membrane where dried down chemistry will produce a color change to indicate the presence or absence of drug. In order to be used in rural malaria endemic settings, it has to be equipment-free, it has to be user-friendly and rapid, and it has to be able to be stored at ambient temperature for prolonged periods of time. We've identified these four small molecule drugs as the most commonly used partner drugs in ACTs, and so these are the 4 drugs that I will be targeting for detection with the assay. So splitting the research goal into my 4 main objectives, the first is to develop a system for blood collection and sample preparation, so a way to isolate plasma in the detection membrane so that you don't need access to a centrifuge in order to isolate plasma and use the assay. Second, to create the drug-specific detection system, so how will you specifically bind and detect and differentiate between the different partner drugs. Third, develop the colorimetric sensor and a smartphone application for analysis. So how will you produce some visible change upon binding of the different drugs? Ideally, it will be a dye or a color-based system that you can see with the naked eye, and you don't need a reader or any kind of equipment to analyze the assay. But we're also working on developing a smartphone application so that the healthcare worker or clinician can take an image of the colorimetric output, and an algorithm will help automate interpretation to remove subjectivity associated with the user interpretation of a color-based assay. And then lastly, we'll test the feasibility and the accuracy of the assay in rural malaria endemic settings, likely in Cambodia, and we'll validate the assay against the gold standard LC-MS methods in patient samples. Okay, so the first objective, the system for sample preparation. I tested 4 commercially available filters for their ability to remove hemoglobin and isolate plasma in a secondary membrane. So 2 desired characteristics that I was looking for in the filter are their ability to remove the hemoglobin or whole cells, red cells, from the plasma. This isn't because I think the red cells or the hemoglobin would interfere with the chemistry of the assay, but the hemoglobin would mask any colorimetric output. And next, to recover the maximum amount of plasma from the sample in the secondary membrane, so that I have more patient sample to work with to improve the sensitivity of the assay. So I applied whole blood to 4 of these different filters and collected plasma that wicked through to the secondary detection membrane to test these commercially available filters. And as you can see, really just one of them, the Pol Vivid GR membrane, was able to effectively separate all of the hemoglobin. It was also the most effective at recovering plasma in the detection membrane, recovering about 70 to 75% of plasma from the original fingerstick sample. The next objective, the drug detection system, I determined that in order to be able to specifically detect and differentiate between these 4 partner drugs, I would need to use either an aptamer or an antibody-based assay. Aptamers are single-stranded DNA that bind their targets via weak electrostatic forces and conformational fits, and the variable sequence of the oligonucleotide determines how it will fold and which targets it will bind to. Antibodies are proteins that similarly bind via electrostatic forces and conformational fits, and they have variable amino acids at their binding regions to determine their specificity for targets. I decided to start focusing on selecting aptamers against these drugs because they can be selected in vitro, and they're inexpensive and easy to reproduce once you know your target binding sequence, and they're more stable than antibodies when dried down. And so I've so far attempted two different methods to try to identify aptamers that bind my target drugs. One, a process called CellX, the in vitro selection process, and then also by making modifications to an existing small molecule aptamer. Okay, so here's a diagram of the CellX protocol. This is the protocol for identifying aptamers to new targets. It stands for Systematic Evolution of Ligands by Exponential Enrichment. And the principle of CellX is that you start with a random library of oligonucleotides, single-stranded DNA or RNA. I use DNA. And you expose this library to your target, so in my case, the antimalarial drugs. And you collect any of the sequences from that library that bind your target, and then you amplify those binding sequences, bring them back via PCR, bring them back to single-stranded, and now you have a second iteration of your library pool, and you bring this through the next cycle. And you continue doing selection cycles until your library pool evolves to contain sequences that bind your target with a high affinity. The sequences that bind your target best are the ones that best survive the evolutionary process and become most enriched in later rounds of selects. And you can also do negative selection in selects. So, for example, if I did selects rounds against lumefantrin, I want to identify an aptamer that binds lumefantrin but doesn't bind the other 3 partner drugs. So at some point in one of my cycles of selects, I can take my pool and expose it to the negative targets, the other 3 drugs, collect any of the sequences that bind those drugs and remove them from the pool, and then continue with my positive selection against lumefantrin. I've done 2 selects experiments against lumefantrin, and after them I sequenced the results from the final pool, so after 7 or 8 cycles, as well as one intermediate pool, and then sorted the sequences based on their abundancy. So if you see in the table on the left, LF1 C3 is the 3rd cycle of the first selects experiment, C8 is the 8th and final cycle, and in the 3rd cycle, the most enriched sequence, so the sequence that appears most frequently, only appeared 3 times, but by the final cycle, the most enriched sequence appeared 141 times. So you see an increase in enrichment of sequences throughout the selects. Then the next step is taking these most enriched sequences and testing their ability to bind your target and finding a way to quantify that binding. So I attempted to use 2 different sort of quick screens for binding to identify sequences to further characterize. One was a gold nanoparticle aggregation assay, and the other was a fluorescence binding assay where the intrinsic fluorescence of the small molecule drugs was quenched upon aptamer binding. But neither of these assays gave me real definitive information on aptamer target binding, so eventually I used isothermal titration calorimetry, or ITC, to measure binding. ITC measures the heat released or absorbed upon aptamer target binding, but it requires a lot of material, a lot of aptamer, and a lot of drug. So with the amount of aptamer that I was able to synthesize and purify in the first round of testing, I tested 5 of my enriched sequences, and in 3 of them, at these concentrations, I didn't detect any binding. In 2 of them, I detected weak binding, and you can see the binding curve on the left. On the right, this is the binding curve with a fit that provides a dissociation constant of 5 micromolar about, which is the low micromolar to high nanomolar range of a dissociation constant is about the range I would like for use in my assay. But I'm in the process of resynthesizing these aptamers so that I can repeat this study and get a better fit and less error in my measurements. So another potential method to identify sequences that bind my target drugs was by making modifications to an already published aptamer that binds cocaine. A group in Canada at York University published their findings that this MN4 aptamer, which is a cocaine-binding aptamer, also binds the small molecule quinine with a dissociation constant of 3.5 micromolar. So I reached out to them to see if they thought their MN4 aptamer would also bind some of the quinoline antimalarial drugs because they share the quinoline group structure. And I sent them some of these drugs, and they tested the ability of their aptamer to bind these 5 shown on the right, the quinoline antimalarial drugs. And they found that this aptamer binds the drugs in the low micromolar, nanomolar range. You can see the KDs in the table. So this was great. It binds all of these drugs with a really high affinity, but low specificity. Obviously, it binds all of the drugs, so you wouldn't be able to differentiate which drug is in a patient's blood. It also binds quinine and cocaine, so really doesn't have the specificity that I would need to use in my assay. So in order to try to improve specificity, I synthesized some aptamers with variations to bases, different lengths of stems of some of the aptamers, as well as changing bases around the pocket in the middle where the drug is expected to bind, and hoping that this will improve affinity for some drugs and remove affinity for others. And I'm in the process of testing these aptamers right now. The third objective, the colorimetric sensor and smartphone application. Really, I'll begin working on the colorimetric sensor once I've identified aptamers that bind to the drugs. But I've started doing some initial testing with the gold nanoparticle aggregation assay. So gold nanoparticles, when they're dispersed in solution, they absorb light at a certain wavelength and appear this pinkish color on the far left. But upon adding salt to the solution of gold nanoparticles, it destabilizes them in solution and they aggregate, and then they absorb light at a different wavelength, and you see a shift to the darker purple color. But it's been found that single-stranded DNA adsorbs to the surface of gold nanoparticles and stabilizes them, preventing their aggregation in the presence of salt. So if you use your aptamer specific for your target as the single-stranded stabilizing DNA, then when you add your target to solution, the aptamers they'll have a much higher affinity for target than for the way that they're weakly adsorbed to the nanoparticles. So they'll leave the nanoparticles to bind target, and then the nanoparticles aggregate. And you see this nice color change here in the presence of target. And I did some, you can't maybe see it so well, but I did some testing with a previously published kanamycin aptamer system, and I also did some proof of concept testing in the ability to dry down the nanoparticle aptamer system and then resolubilize it by wicking sample through with target and causing aggregation. So in the paper sample sort of in the middle of your screen, you can see a negative, it's the pinkish color, and then a solution with 10 micromolar, so pretty high concentration kanamycin. You can hopefully see that the particles were resolubilized and aggregated with kanamycin present. But obviously I have a lot more optimization to do around this sensor system, and I'd like to look into other systems, like using the peroxidase activity of the nanoparticles to enhance the readout, and also tagging the aptamers with dye molecules as another sensor method. And with help from Jay, a master's student in the Department of Computer Science, we started developing the framework for a smartphone application that will walk the user through enrolling a new patient and entering some information about the patient, and then the app will instruct the user how to apply sample to the device. It will have a timer to time how long the device should run for, and then indicate when the device will be ready to be imaged for analysis. So we have the code for this user interface, and once I've worked out and optimized the colorimetric sensor, we'll start working on the algorithm development for the automated analysis of the image. So my immediate next steps are to continue the affinity testing of the sequences that were the result of my CellX against lumefantrine, and also to continue testing the cocaine binding aptamer variants via ITC. I also want to repeat CellX against lumefantrine and decrease the concentration of lumefantrine that I used in my positive selection, so I can try to isolate sequences that bind at a lower concentration. I'll do more cycles of CellX next round to try to improve enrichment, and I'm going to make some changes to some of the purification methods I used. I'm also going to start looking into the potential of an immunoassay and using monoclonal antibodies in case I can't identify aptamers that bind these drugs. So commercially available antibodies for chloroquine and amidiaquine already exist, and I'll start looking into what it would take to develop monoclonal antibodies against lumefantrine, mefloquine, and piperacin as a backup plan. And I'm going to continue working on the colorimetric sensor development. I'm headed back to NIH next week, where I'll continue doing this. So I want to thank my supervisors, Rick and Martin and Joel, and Jay, the master's student who's done all the coding for the app. He's really great. Everyone in the Tom Brown group in the chemistry department here who I'm working with right now, especially Erin, Afaf, and Pui, and Slajana from the group at York University, and my funding from the NIH Oxygen program. That's it. Thank you.